We can get started. So it gives me great pleasure to introduce our P30 seminar speaker today. Dr. Jesse Buckley is an associate professor of environmental health and engineering and epidemiology at the Johns Hopkins Bloomberg School of Public Health. Her research aims to characterize environmental exposures during early life and determine their effects on child growth and development with the goal of informing policies targeted at improving child's health. As a recipient of an NIEHS ONES award, she is studying the impacts of endocrine disrupting chemical mixtures on child bone health. In addition, Dr. Buckley serves as Director of Chemical Exposure Methodology for the ECHO Data Analysis Center at Johns Hopkins and is co-chair of ECHO's Chemical Exposure Working Group. Dr. Buckley earned her MPH Environmental and Occupational Health from the George Washington University and PhD in Epidemiology from the University of North Carolina at Chapel Hill. So I nominated Dr. Buckley to speak with us today because her name is pretty much inescapable when it comes to children's environmental health. And her work is really at the forefront, spanning across chemical mixtures, chemical exposures, and child health outcomes, as well as in the development of statistical methods for mixtures analyses. And so I really enjoyed getting to speak with Jessie earlier this morning, and I'm really looking forward to her talk with us today. And so with that, thank you, Jessie, for joining us.
Thanks so much for the invitation. It's always nice to even virtually try to meet some other people. And I'm excited I was asked to talk today about our work in ECHO. So today I'm going to be basically talking through a multi-year sort of strategic planning effort that many of us in ECHO undertook to try to decide what kinds of chemical exposure and health outcome opportunities there are in this large NIH-funded program that spans 70 cohorts and 50,000 mother infant pairs, and how we can best leverage the resources in ECHO to understand how these exposures might affect children's health. And I just want to start off also by acknowledging Tracy Woodruff at UCSF, who was my sort of partner through this work and who has done an incredible job helping me understand how to navigate a large program like this as well. So I don't really have to tell this group very much about industrial chemical exposures. We know that they're ubiquitous, they're in consumer products that we use every day, and that there's about 30,000 pounds of industrial chemicals produced per person per year in the US. So we're all kind of walking around in this chemical soup. And specifically regarding pregnant people in the United States, 43 chemicals in NHANES were found to be in every woman who was assayed in this study in earlier 2000s. And notably, it's important to recognize that these chemicals occur in mixtures. And so you can see in this figure, each bar represents a participant, and every person in the study had at least 25 chemicals detected in their either blood or urine. So during pregnancy, we have very ubiquitous exposure to chemicals. And then it's also well known that chemical exposures during critical periods of development can and do affect the development of the fetus and their health outcomes during childhood. So this is sort of the background. Then we have this new opportunity with the Environmental Influences on Child Health Outcomes, or ECHO program, to try to understand how chemical exposures can affect children's health. And so today's story is sort of about how our working group in ECHO tried to address this problem. And just as a really brief background for those of you who may not be familiar with the program, ECHO is a nationwide NIH-funded study that is comprised of 69 longitudinal pregnancy and birth cohorts from across the United States. We have data from over 55,000 mother-infant pairs from these cohorts, and it's comprises extant cohorts that have already recruited participants, and then funds ongoing follow-up of these participants. And we also have a really wonderful opportunity to have a supplement with the Human Health Exposure Analysis Resource, or the HERE program. So there were also funds available to add new chemical biomonitoring in addition to whatever chemical biomonitoring was in place when ECHO cohorts kind of came to the program. So we wanted to be able to help achieve ECHO's overarching scientific goal to answer questions about a broad range of early life environmental influences on child health and development. So the program was funded in 2016, and shortly thereafter, there was the establishment of ECHO's Chemical Exposures Working Group, which is the group that I currently co-chair along with Debbie Bennett, and Tracy Woodruff was a former co-chair with John Meeker. And so from the very beginning, we wanted to try to understand what kind of data we have in ECHO from the cohorts that were joining ECHO who already had chemical exposures, and try to figure out what the best way was to leverage this existing data for chemical exposure studies. But we also wanted to recognize that there's a lot of chemicals that maybe hadn't been measured by ECHO cohorts or haven't been measured writ large in the US population, and understand how we might be able to fill some of those important gaps. And then together, kind of combining the existing data and new data, create a platform for solution-oriented research to advance children's health. So regarding the first point of trying to understand what kind of data we have in ECHO, I'm just going to briefly talk about this because it might be of interest to people who want to collaborate. We did publish a paper that reviewed information on chemical exposures, both biomarker data and questionnaire data related to exposure sources, that was published a couple years ago. And what we found from this was mainly that there were a good number of ECHO cohorts that have extant data, biomonitoring data in both pregnant people and their children. So this bar chart just kind of shows the number of cohorts that had existing biomonitoring data for various chemical classes, either from mothers during pregnancy or from children. And we gleaned from this that ECHO cohorts already had a lot of data on environmental phenols, phthalates, metals, and tobacco biomarkers. And so that was a good place to start with launching studies that could get going right away and focus on leveraging this data that already existed. And then we made some recommendations about, now that we kind of knew the landscape of what chemical exposure data already existed, the kinds of studies and the kinds of chemical classes that we thought should be biomonitoring using the HERE supplement that ECHO had. And our main kind of conclusion from this was that we should be focusing on emerging and novel chemicals. Those were recommended for biomonitoring both in the prenatal and postnatal period because we don't have a lot of data on those. And so this review paper really focused on chemicals that are measured in NIEHS and that a lot of the cohorts already had measured using targeted assays. As we all know, NIEHS measures only about 350 chemicals, and there are more than 40,000 chemicals actively used in the United States. And 8,000 of these are high production volume chemicals. And so while it's important to keep studying the chemicals that we know about, there's an important gap that we wanted to try to fill with relation to trying to understand what exposures to pregnant women and children are, and also what their health consequences are for these chemicals that we don't know very much about. And so that's what I'm mostly going to be talking about today, which is our kind of process to identify, measure, and study novel chemicals in ECHO. So I'll discuss how we came about recommending chemicals for biomonitoring, and then the pilot study that we just completed, where we measured some of these novel chemicals in pregnant women in ECHO. And then we've launching a full-scale study to assess these chemicals in a large sample size of women in ECHO. And then we'll be looking at lots of different child health outcomes once we've created this database that is very large and unique. So the first step was to identify and prioritize chemicals that haven't really been looked at before in most of the assays that have been done in ECHO cohorts or really in the US at all. And so this is a paper led by Ido Palazzari and Debbie Bennett, where it was published in EHP a couple years ago, where we tried to kind of narrow down a landscape that we would be able to recommend and focus on. I'm just going to very kind of quickly go through the process that we used to identify chemicals for biomonitoring. And we started off with a really large number of chemicals from various databases that were kind of our top level. We had over 170,000 chemicals that were included in this first initial screen. Then we narrowed them down to 932 chemicals where they were found to be quantifiable in environmental samples, or the chemical structure suggested that they were likely toxic. From there, we grouped them into 5 groups and decided to just sort of set aside chemicals that were measured in NIEHS because we had already focused on those. And we set aside legacy chemicals where we knew exposures were going down because those weren't really thought to be something that we should focus on moving forward with ECHO. It's left us with 3 groups of chemicals that what we did was sorted them into 8 chemical classes that we felt we could focus on in one paper, but there's lots of chemicals left for us to look at in future work. So these 8 chemical classes are listed here, and we went through a process to take 155 chemicals and determine which ones seemed to be the most important with the most data that we could use to suggest that probably chemical exposures are occurring and that these chemical exposures may be toxic. So the first step was to see if they were present in human biospecimens or environmental media. And if so, then they were continued through the process. And if not, they were either deemed low priority for ECHO or deferred pending further data. And then we looked at whether or not they were, there was evidence that they could be toxic. And again, they were either classified as low priority, deferred, or potentially high priority. And then we determined from the literature whether or not there seemed to be an available biomarker, so that would be something that we could actually measure. If they sort of hit yes across all of these boxes, then we consider them to be a high priority candidate for biomonitoring. So I'm just going to very quickly summarize the process with one example here. This is the organophosphorus-based flame retardants, or OPFRs or OPEs, depending on your favorite acronym. So these are several OPE chemicals that were selected for our process. And just as a spoiler, the orange ones are the ones that we ended up recommending for biomonitoring and ECHO. So the first step was to look at whether there was evidence that they were present in biospecimens or environmental media. And you can see the gray boxes suggest that we did find evidence of this, and then the numbers are the citations that are found in the manuscript. So for many of these, we did find that they were had been measured in biofluids, air, indoor dust, or food and water. After that, we then looked to see if there was a concern for toxicity. And with this, we use in vitro and in vivo studies, as well as HTP assays and QSAR modeling, and searched to see if there was evidence of toxicity specifically related to the kind of priority outcome areas of ECHO, which included endocrine, developmental, reproductive, and neurotoxicity and obesity outcomes. And then we also included carcinogenicity, just because that is another important outcome. So here we found a fair amount of data for many of these chemicals indicating especially developmental and reproductive toxicity. And then the last thing we did was to see in the literature if there was an available biomarker. And again, here it seems that several of these had been measured in hair, fingernails, urine, or breast milk. So at the end of this process, we took these, went through the same process for every of the 155 chemicals in our list. And you can see here, we, in the end, recommended 36 for biomonitoring, and 108 were deferred. And most of these were deferred because they lacked information on either toxicity or an available biomarker. Those that didn't have an available biomarker were sort of, some of those are carried forward into the work that I'll keep discussing because we worked with a laboratory to develop biomarkers. And then 11, we kind of found good enough evidence to suggest that they're a lower priority for biomonitoring. So most of them really lack the data that we needed to consider them to be high priority. These 36 are the ones that we focused on. So then taking this kind of forward to the next step, one of our co-authors on that paper is Dr. Cannon at the NYU Wadsworth Laboratory, and that is a HERE laboratory that has an ECHO supplement. And so through working through that process with that paper, he had already been kind of planning a multi-class assay that would be able to measure multiple chemical exposures across several chemical classes in a single urine sample. And so what we did was collaborate with him to add some of the chemicals that we had kind of designated as high priority for ECHO to this assay. So we were really lucky to be able to be in the right place at the right time for that. And so he developed this assay. It measures 89 urinary biomarkers from actually 103 chemicals and across 9 different groups. So normally in echo, cohorts have measured some phthalates or some OPEs or some insecticides, but they're all kind of in separate panels and requiring multiple urine samples to do each of those panels. So this is just one assay using 0.5 ml of urine. So it's really cost effective and yields a lot of data from a very small urine sample, which was really attractive to the ECHO program. So then we wanted to take this assay and apply it to participants in ECHO. So our first step for this was to conduct a pilot study, which would apply this assay to samples to make sure that some of the that these chemicals are indeed detected in ECHO samples and to justify better biomonitoring within the ECHO program. So for the pilot study, we were lucky to work with 9 cohorts across the United States from different areas, and one of them is Fair Start from working with Julie Herbstman here at Columbia. And so we got around between 15 and 20 samples from gestation from each of these 9 cohorts. So we had 171 participants in this pilot study. And we really tried to capture variation across the United States and also in sort of temporal and sociodemographic diversity. So you can see here we had a maternal age of about 30 was the mean. And then we had 34% non-Hispanic white, 20% non-Hispanic Black, and about 40% Hispanic participants in this pilot study. And there was a range of educational attainment with about 46% having a bachelor's degree or higher. The urine samples were also collected kind of across time, and we had some samples from before 2015, and then most of the samples were taken between 2016 and 2020. And so, as you recall, there were 89 analytes in this assay, and we detected 45 of them in more than half of the urine samples. And so this represented exposures to chemicals in multiple different classes. You can see in this, I know it might be a little bit difficult to read, but we have exposures representing both kind of legacy or kind of known current use chemicals. For example, for the bisphenols, we measured bisphenol A, but we also had replacements such as bisphenol S. Similarly with the phthalates, we measured many phthalates that are included in NIEHS, but we also included things like terephtalates and DINCH, non-phthalate alternative. So we definitely found common and ubiquitous exposures to many of these chemicals, and I'm just going to focus on 3 representative ones. So one of the things that was striking to us is that we found neonicotinoid pesticides in almost all of our samples, and N-desmethyl acetamiprid and thiamethoxam were found in almost all the samples. And these are kind of neuroactive insecticides that are chemically similar to nicotine and have been kind of in the news based on their effects on bees and et cetera. So this was an important finding and something that we really want to follow up on. We also found, and I probably should describe the box plots here. So the first, the blue box is the overall sample, and this is the concentration in everyone, and then each of the subsequent boxes is a different cohort. So I'm sort of organized from the West Coast to the East Coast. So we have our 3 California cohorts, and then we had kids, which is in Illinois, the Atlanta cohort of Emory in Georgia, and 3 cohorts in New York, or 2 cohorts in New York, and then New Hampshire and Puerto Rico. So you can see that regardless of the location, we found these pesticide in most of the samples. Similarly with BPS, we found them pretty ubiquitously, and I think it was really interesting that bisphenol S is used as a replacement for bisphenol A in plastics and food, beverages, and can linings, and it's becoming of concern because it could be a regrettable replacement for BPA given its structural similarity and estrogenic effects. So we found bisphenol S in more samples than bisphenol A, which was really interesting to see. We also measured a number of phthalates. Many of them were ubiquitously detected in our study population. This one is one that has not been measured in human samples previously, monohydroxyisodecylphthalate, and this was pretty ubiquitously detected across all of our samples. And there were several others that were also very commonly detected that had not been measured previously. Another important point about these data is, we have the ability to measure many chemicals in the same people, and we found that, indeed most of these were detected or 45 of them were detected over half the population, and there's evidence that, these women were really exposed to exposure mixtures. Here you can see the Spearman correlations across classes, and this is for the analytes that were detected in at least 70% of the pregnancy urine samples. As you might expect, there's higher correlation within class, particularly for the phthalates. And it's an important finding because when we think about looking at these exposures in future health studies, some of these correlated exposures need to be accounted for, either in terms of co-pollutant confounding or potential joint effects. And so that's a really important piece of our future work. Then we have the associations. We looked at associations of each of these chemicals with different demographic and characteristics. And so one of the things that we found that was really notable to us was the time trends. And so here you can see that many of these analytes either increased or decreased with time. For example, with the bisphenols, as you might expect, we found increasing concentrations of bisphenol F and bisphenol S with calendar time. And we found decreasing concentrations of many of the phthalates over calendar time, at the same time increasing concentrations of bench, which is a phthalate alternative plasticizer. And this is kind of mirroring some of the findings that have been shown in other studies and NIEHS. The other thing that we found that was really important is differences in the concentrations of these chemicals by race and ethnicity, and particularly many of the chemicals we found were higher among non-Hispanic or the Hispanic participants in our study. And this was pretty across the board. These relationships are unadjusted given our sample size, but we also looked at adjustment for socioeconomic status and maternal age and calendar time. And we restricted to cohorts, not including the Puerto Rico cohort, and their findings were pretty similar. And then this is not part of the paper because it's very exploratory and unadjusted, but because we wanted to demonstrate to the ECHO program that these chemicals are important for health. We also just kind of explored some associations with birth outcomes. So this is a volcano plot showing the difference in gestational weeks associated with increased exposure to or concentrations of each of these chemicals. And you can see this one of the neonicotinoid pesticides and many of the phthalates were associated with lower or shorter gestation in unadjusted analyses. And similarly, some of the phthalates were associated with lower birth weight. So this is just exploratory at this point. It definitely demonstrates that probably more work is needed to understand how these chemicals might be related to health in our study population. So with that, just kind of to summarize the pilot study, we found widespread exposure to multiple chemicals and pesticides, and was declining exposure to chemicals that have been the focus of policy actions or market-based campaigns. So we saw that there were lower exposures to phthalates during the study period, and also higher exposure, though, to their replacements, which is really concerning, given that we know less about exposures to these replacements and less about their health effects. Many of the analytes that we looked at were positively associated with Hispanic ethnicity in our study, and we found sort of suggestive associations that some of these chemicals may be related to birth outcomes. And so what we really take away from this is that we need to do a larger study to look at associations with birth outcomes and other health effects. And if these findings are replicated, then some of these chemical exposures could be underpinning the transgenerational propagation of health disparities, these higher exposure and higher vulnerability that can be ongoing, and if we aren't able to do something about these chemical exposures. The next thing we're doing in ECHO is to identify novel chemical exposures in a larger sub-study. So for this, we've designed a study that was approved by ECHO and now is underway, where we're going to use this multi-class assay to measure these novel chemicals among over 6,500 pregnant women from 20 cohorts. So this is going to be a much larger study. The first step in the analysis will be to look at associations with perinatal outcomes, such as low birth weight, preterm birth, and small for gestational age. But in addition to kind of understanding exposures to these chemicals and their health outcomes, we also have added a couple of sub-studies to help us understand the exposures, and particularly because we're measuring these chemicals in urine, and many of them likely have a short half-life. We're doing a sub-study to determine the intra-individual variability. Since we are going to have just a single measurement for most people, we wanted to understand how variable they are over time during pregnancy. And we can also use this information to do sensitivity analyses to correct for exposure misclassification in our larger study. In addition to that, we're also doing a sub-study looking at paired samples from mothers and then their children after birth to see if there's correlations between mothers and their children in terms of their exposure to these chemicals. So this is going to be a really large undertaking. It's been a long of time in the making. And there are a couple of lessons we also learned that I don't have on the slide here, but many of the chemicals that we tried to measure, we measured in urine. And so for many of the pesticides, for example, that we wanted to measure, we weren't really able to find, we either didn't know what their metabolites were, or we couldn't find a standard for the metabolite. And so some of the chemicals that we really wanted to include in this, we weren't able to quantify. And in addition to this work, we're wanting to do some additional studies using blood or being able to get at some of the other more persistent chemicals and not just the non-persistent chemicals. Another thing we're doing that I don't have on the slide is one of the chemical classes we focused on in the original paper was aromatic amines. And we did a parallel sub-study to the one that I just presented that's under review now, where we quantified exposure to aromatic amines and melamine and cyanuric acid. And we also found ubiquitous detection of those chemicals in pregnant women. So we have another study that's in a smaller subset of 1,500 women to look at those chemicals. So we're really trying to build up a platform where we've been able to characterize exposure to these chemicals that we aren't often looking at in other studies, and be able to link them to child health outcomes. And so with this platform, we're starting off with pre, peri, and postnatal outcomes, but in ECHO, there's a lot of data on the other ECHO focus areas, which include airways, obesity, neurodevelopment, and also positive health, or kind of the absence of health problems. And so when we are able to combine all of the extant data and all the new data that we're collecting, it's going to be a massive resource for researchers, both in ECHO and outside of ECHO. There's going to be a public platform that investigators can use to propose their own studies, sort of similar to NIEHS. And so there's just a lot of opportunities to build on this resource and collaborate on these studies. And I am, really happy to hear from people who might want to leverage these data for new questions. There's only so much time in the day, and there's just a real abundance of data here. So please do reach out if you have ideas. And so kind of to wrap things up a little bit, we found widespread exposure to multiple chemicals during pregnancy, and many of these we measured for the first time in human samples. There's a real unprecedented opportunity to use these data to understand chemical exposures and health effects. And then another area that we would really like to do more work in is to study where these chemicals might be coming from. So because of the lack of labeling requirements and just like a real difficulty in understanding where sources are. There's a lot of information in ECHO to also look at potential sources, whether it be dietary route of exposure or household characteristics, et cetera, and be able to understand where the chemicals come from so that we can inform solution-oriented research and interventions to improve child health and reduce health inequities. And hopefully, combined with the exposure and outcome data and then understanding the sources, we'll be able to inform these in exposure reduction strategies that can focus kind of across the range of different types of either individual, household, consumer, and kind of national level policies to reduce the burden of these chemical exposures in children's health. Again, I just want to quickly acknowledge Tracy Woodruff and then also Jordan Kuiper here at Hopkins, who did most of the data analysis, and Dr. Cannon at NYU for his leading his laboratory team to really develop the assay that we used, as well as to apply it to our samples. And now, like, going through the 6,500 samples that we have waiting in his freezers to do the larger scale study, as well as all the different people on the writing team and the members of ECHO's Chemical Exposure Working Group and the study participants, as well as, of course, our funding. And I went by pretty quickly, but I wanted to leave time for questions and to share any other information about ECHO that you might like to learn more about.
Thank you, Jessie. So yeah, as Jessie mentioned, we have time for questions. So I think Andrea has a question.
Yeah, first of all, thank you so much for this presentation. It's incredible to see both the amount and quality of work that the consortium has done, and congratulations for leading these efforts. I think I have two questions. One, I was very impressed with the panel of 89 chemicals that has been developed and used. So I'm wondering whether there is any trade-off between measuring so many chemicals at once and the precision or sensitivity of the assays, and where that puts us compared to studies that might focus on a smaller panel of chemicals that are more focused. And the second, yeah, please.
Oh, okay. Yeah, that's something that we've talked a lot about, particularly because there's obviously the cost effectiveness of measuring all of these, but the trade-off is the sensitivity. So because most of the chemicals that we studied were so ubiquitously detected, we didn't need an assay that was super sensitive at the low levels. And so we felt that, the ability to measure more chemicals was worth the trade-off. Some of the cohorts have measured things that have already measured in the same samples, either like a different assay of phthalates or bisphenol. So something that we could do is compare the concentrations in our subsample that were measured in the two different assays. But in general, the limits of detection were really similar to the NIEHS limits of detection, and so we didn't feel like we were sacrificing very much.
And do you have data on precision? I mean, what is the sample?
Yeah,
yep.
It varies, definitely varies. So for some of the chemicals, so we did actually a lot of work on that with blinded duplicates, and we found that the precision for chemicals that were closer to the LOD, there were some high CVs for sure, especially with low concentrations, as you might expect. But then for the chemicals with higher concentration or more variability or higher levels, the CVs were lower. So another thing was that we knew we were trying to measure some of the chemicals we were trying to measure, we knew we probably wouldn't detect in urine anyways. So we kind of attempted it. And for those, the precision was not very good, and they were also like low detects. And
always related to this panel you all developed, it seems that the process brought you to focus on or come up with chemicals that in a way are very well known in the field, and there already have been many studies on. And at some point you had a slide on new chemicals, but I'm not sure I understood the plan of the ECHO consortium to evaluate emerging and new chemicals. I would love to hear more about that if you have any information to share.
Yeah, so you're right that many of the chemical classes that we focus on are chemical classes that are well studied, but we try to include a lot of of the replacements. So in addition, so I think I probably went by really fast, but we did measure things like bisphenol A diglyceryl ether, like badges. We measured many phthalate alternatives, and a lot of them just weren't detected, which is good news, I guess. And so that was something that we were kind of surprised to see. Not that we wanted to see high exposures to some of these replacements, but they were included. We included 4 or 5 different benzophenones, whereas NIEHS only measures benzophenone 3. And so that was one kind of step. And then what I didn't present on today was the data on melamine and cyanuric acid, and we measured 18 aromatic amines. So those required different assays because they couldn't be reliably run in the same multi-class assay. And we also found some kind of really interesting and ubiquitous exposures to, I think, 6 of those aromatic amines. And some of them were highly correlated with cotinine or nicotine concentrations in the samples as well. So that's another paper that we're working on now. And I also agree that a lot of the chemicals that we kind of would want to focus on would be better to measure in blood. And so that's going to be a whole nother process.
Like to say, as a follow-up comment on my first question, I guess, even if precision is lower than in more targeted assays, being able to do it on a sample size of 60,000, I think, overcomes any loss of precision. Perhaps it's an interesting calculation to do if you had accurate data, but I guess, if you can save money and be able to scale up to 60,000 samples, that there's no better way to compensate for precision than having a sample size like that.
Yeah, that's definitely one of the things that we're considering. Yeah.
Thank you so much. Really impressive, and glad you had with us today.
Yeah, thanks.
I also had a couple of questions. These are more structural questions. So in your pilot, you showcase this really nice way where you are nominating emerging chemicals for which there's no existing biomarkers, and then you can pull in the expertise from here to develop those biomarkers and then implement in this echo study. And I was wondering how much in general there's this built-in crosstalk between here and echo to do these types of things, outside of this pilot.
I think there's a lot of that crosstalk. I think it's been one of the most valuable pieces of being part of the program for me, is being able to get to know the Lab Hub PIs. So with the initial, right when we launched, we immediately started this chemical exposures working group with the Lab Hub PIs being part of that group. And so Dr. Cannon was part of the group that did this review paper, and he got really excited. I think that's maybe part of the reason that he developed the aromatic amines assay that he worked on. And so having his input, and it was his suggestion that he could add a bunch of these chemicals to a new assay that he was already working on. And so having these conversations happening throughout, kind of us telling them what we're working on and then them telling us what they're working on, has been super helpful. And it's the same thing with some of the other Lab Hub PIs, like Patrick Parsons is working on some interesting metals assays and different matrices. And there's some other pilot work that were funded by ECHO. So if we wanted to try to measure certain chemicals in placenta samples, and the Lab Hub didn't necessarily have the capability previously, now we're sending ECHO samples and we're doing pilot studies. So I think that's how we're trying to kind of increase the innovation instead of just relying on kind of the data we already had.
I had one other question related to sort of infrastructure. So I guess the overall goal for ECHO, as I understand it, is to build a sort of synthetic cohort across these individual existing cohorts. And I'm wondering about, all of these cohorts bring different data sets in. And so if you're an outside investigator and you have a specific question and you want to leverage multiple cohorts, and there's maybe, especially for some of us who are working more in the biospecimen biomarker space, how feasible is it, or is it feasible to go beyond just existing data that's already collected? For example, placental epigenetic data is collected maybe on just 1 or 2 cohorts or just a few cohorts, and you really want to get to this sort of synthetic nationwide analyses. So an echo-wide, ideally something spans across many cohorts. Is it really logistically feasible to propose outside of your own R01 funded mechanism to get at that kind of level of echo-wide analyses that could be proposed in that way?
Yeah, that's a great question and an interesting challenge. So I mentioned that ECHO has a HERE supplement. So for targeted and untargeted chemical assays that can be done by the lab hubs that are funded through ECHO, you can just propose something. Right now, they're still accepting applications, and that's how a lot of this work kind of started. You can even use samples that were collected pre-ECHO, so they don't have to be ECHO samples. At the same time, those samples are available based on the whether or not those cohorts collected samples before ECHO. So, and whether or not they want to share them. And they're under no real obligation to share them. At the same time, ECHO is requiring new biospecimen collection. So children's samples are being stored in a repository, and you can apply to use those samples. If you are trying to do chemical exposure analysis, then there are funds for that. If you're trying to do other kinds of analyses and you have funding, then you can apply to use the samples.
Great, thank you. Are there any other questions? So I see that Ike had posted a comment. She was really interested in your neonicotinoids findings, and she spoke to some findings in bees as well that indicate toxicity. So I want to speak to that. So I don't see that there are any other additional questions. So I really want to thank you for your time, and thank you so much. I learned a lot about all the work that you're doing in ECHO. So thank you.
Thanks for having me. And if people want to learn more about ECHO, especially if you're interested in chemical exposures on your gal, I'm always happy to answer questions by email or follow up. Or if anybody's interested in kind of leveraging the data that are being created now, these assays should be done in December, and it's just going to be, getting people involved, and we would love to have more people come on board.
Wonderful. Right. Thank you. Take care.